Dr. Wargo is from MD Anderson Cancer Center, and she will share with us her quite also groundbreaking work in utilizing the microbiota as another adjuvant for immunotherapy, particularly in the context of melanoma. So with that, welcome.
Thank you very much. It's a great honor to be here. Sorry I'm a little bit late. We had a flight delay this morning, but I'm here nonetheless, and honored to be here. Really, thank you to the organizers for the kind invitation to be here. Thank you to all of you for being here and for caring about this. And I'm a professor of surgery. I'm a surgeon scientist at MD Anderson Cancer Center. I've been there over a decade. And so what I wanted to do today is to share a little bit of insight onto how we can target the microbiome to promote health and to end cancer. And so before I get started into the talk, though, I wanted to really kind of spend a little bit of time to thank everyone who's contributed to this work, and that first and foremost includes patients and their families, also all care providers. The MD Anderson, we have a moonshot program, which really kind of helped fund all this work. Also to research teams in Prime TR, which is our microbiome center there. And then to the leadership at MD Anderson, amazing people, and supporters and collaborators worldwide. And thank you to all of you for really what you do to promote health, and really for those who study cancer, for what you're doing for patients with cancer. It's truly been transformative. And so as a surgeon scientist, I know my limitations. I always say never overestimate the power of a scalpel, and we know that treatment with surgery is just one part of how we can cure patients with cancer, but immunotherapy is a particular form of treatment which has totally revolutionized cancer care, okay? It's actually made its way as one of the very pillars of cancer care. This was named the Breakthrough of the Year in 2013, and my friend Jim Allison won the Nobel Prize for this in 2018. And I show up here Sharon Belvin, who I can show right there, who was the first patient cured from melanoma. So young woman, had metastatic melanoma, spread throughout her body, got a dose of CTLA-4 blockade, and actually was cured and can now spend time with her kids and is healthy and doing well and loving life. She actually attended the Nobel ceremony with Jim, as did I, a couple of years ago when we went. And now many patients are being cured, okay? And I can tell you that we're using this consistently. I can tell you just last week I was in clinic seeing patients, treated 4 patients who had advanced disease with immunotherapy prior to surgery. I took them to surgery, and when the pathologist looked under the microscope, there was no tumor left. So let's have a little round of applause for that. So pretty dramatic. And I can tell you, I would not have seen that when I started my career over a decade ago, or over 20 years ago now. But what I would argue, though, is that there's still a critical need to improve outcomes to cancer therapy and to limit toxicity in patients with established cancer, as well as opportunities to prevent cancer altogether. And so and of course we've heard about this already, through translational research, we can actually gain insights into factors that actually impact tumor growth and response to cancer treatment that we can actually target. I've spent my career looking at factors that influence response to treatment with immunotherapy and other therapies. And what we know is that there are factors within the host, things like within the tumor microenvironment, like the immune cells, certainly host genomics, systemic immunity, systemic factors like sex hormones play a big role, and then the microbes that are within us. But this also includes factors that are external to the host, and a topic of this conference, really the exposome, if you will. And this includes things like environmental exposures, psychosocial factors like stress, makes a big difference, lifestyle factors like diet, exercise, and we'll talk a lot about that, and also the microbes that surround us. As Barbara just mentioned, the microbes aren't only within us, but a lot of microbes are around us as well. And so really kind of going back a couple of years, after studying response to immunotherapy for a while, we really asked the question, well, considering that humans really do represent a complex ecosystem, what is the role of microbes that coexist with us on cancer and on therapy response? And so, this is no surprise to this audience. So 3.5 billion years ago, right, microbes actually helped us shape the earth for future forms of life. They give us the very oxygen that we breathe today. And now we know that 3.5 billion years later, microbes are not only pervasive in our environment, but also within living organisms. Okay, so take a human, okay, 20,000 human genes, right? 1% of our total genomic content is human. We have between 2 and 20 million microbial genes, okay? 99% of our total genomic content is microbial. And importantly, this is inherently modifiable. And advances in next-generation sequencing, like Barbara just spoke about, is really what has really enabled us to better understand who these bugs are, what they're doing, and how they can impact whether or not cancers develop and how they respond to treatment. And so the importance of the microbiome certainly is in health and disease is increasingly evident, and this very much includes in cancer, where microbes within the tumor and surrounding tissues, as well as microbes within the gut, importantly, and other niches, can actually impact whether or not cancers develop, how they respond to treatment, and then also other physiologic processes. So I pose the provocative question, and I think everyone will kind of embrace this. Could microbiome targeting become the next pillar of cancer care, right there next to immunotherapy, with strategies to monitor and modulate the microbiome to treat, intercept, and maybe even prevent cancer altogether someday? So let's talk a little bit more about this. So before, people often ask me, Jen, how did you get started to study the microbiome, right? And it was actually somewhat serendipitous for me. So I was actually a surgeon back at Mass General Hospital. Sorry, the slides are playing themselves a little bit. And we were actually studying response to treatment with different forms of therapy. And what we found is that we actually studied in patients with pancreatic cancer, we actually identified that there were actually bacteria within tumors of patients with pancreatic cancer, and that these were breaking down gemcitabine into its inactive form, so they could actually break down chemo into its inactive form. We visualized these microbes within the tumor cells themselves, and actually took this back to a mouse model where we treated mice with chemotherapy alongside treatment with antibiotics to get rid of these bugs, and actually cured all the mice of their tumors, and actually published these findings in Science in 2017. And I really posed the question, could we actually use these microbes to predict, outcomes, and maybe even to prevent cancer altogether? And I think the answer could be yes. And if you think about it, there's a precedent for this. Think about HPV, okay? And could we actually mine microbial signatures in tumors, identify that next HPV, and actually prevent cancer altogether? Now that's bugs within tumors, but we know that in addition to microbes within tumors, microbes within the gut can actually profoundly impact our immunity and immunotherapy response. We heard a little bit about this from Elton. And we know that all along this long alimentary tract and vast surface area with all those villi, on one side you have all these microbes, right? And on the other side, you have this dense network of infiltrating immune cells. And so there's this constant to and fro dynamic interaction and candidly, an education of those immune cells at the level of the gut. And so at MD Anderson, we actually studied this in patients with cancer, and we showed that patients who responded to treatment with immunotherapy actually had a more diverse microbiome. We know that diversity is good in all aspects of life. Certainly we showed that. We also showed that patients who responded to treatment with immunotherapy actually had different bugs in their gut. Patients who responded had a higher relative abundance of things like Faecalibacteria, Clostridiales, and Ruminococcacea, whereas patients who did not respond to treatment with immunotherapy had a higher abundance of things like Bacteroidales. And so really, what has this meant for all of our patients? Well, it means, can you use the gut microbiome as a biomarker, and that's something we're testing now. Another thing is, can you actually modulate the microbiome and make patients respond better, right? And so we published these findings in Science with a trio of papers with two other groups in 2018. 3 years later, worked with a group from Israel and published, they actually were back-to-back papers in Science, showing that if you take patients with metastatic melanoma, deadly form of skin cancer, who were treated with immunotherapy and did not respond, their tumors actually grew. If you wipe out their inherent microbiome, transplant in a fecal transplant from a complete responder donor or a partial responder donor, you can actually get those patients to respond to treatment, right? Proof of principle, it can actually work. Fecal transplant can work for patients with cancer, okay? And we also showed, and again, this was back-to-back papers, one from Pittsburgh, actually, from Pitt, and one from Israel, that showed that you could use this approach in patients with cancer. And so really important, right? And transformational work. And we're now working with others worldwide to target the microbiome to better treat, intercept, and prevent cancer altogether using a number of different strategies. And so how can we do this? Certainly, we're still using fecal transplant. It's a relatively crude way, and candidly, choosing the right donor is really difficult. We don't know who the right donors are. Can you use a healthy donor? That's been suggested, versus a patient with cancer who's been cured. Diet and pre or postbiotics, like Aran Elenov spoke about, and also Selden did too. I think I'll show you a little bit of data on how diet actually matters quite a bit. And then certainly microbial consortia and probiotics, but I think Aran and I share a healthy kind of aversion to over-the-counter probiotics, and I'll show you a little story on that as well. And so, but you can use a number of different strategies, but these are potentially, living drugs, at least these two, and it's really complicated to use these approaches. Now, this does include strategies to enrich, favorable microbes and to deplete unfavorable taxa in the gut and also in the tumor of patients with cancer. And so several studies we have undergoing at MD Anderson include Florencia McAllister actually showed in patients with pancreatic cancer that a large percentage of the bugs that were present in the tumor actually were potentially derived from the gut. She also showed that if you took a fecal transplant from a long-term survivor with pancreatic cancer and put it into a mouse that had a pancreatic tumor, the tumors grew slowly and had a really robust immune infiltrate. And she's actually now using fecal transplant to treat patients with pancreatic cancer in the pre-surgical setting. So really interesting, and she's seen some pretty dramatic responses. In addition to this, can you use this in other cancer types? And so Mike Overman at MD Anderson is using fecal transplant in the setting of patients with colon cancer who have progressed on treatment. This is in the context of microsatellite unstable colorectal cancer. If they progress on anti-PD-1, he actually inherently wipes out their inherent microbiome, gives them an FMT from a complete responder donor, and he's seen brilliant responses in that. And then you can actually use this to treat toxicity as well. And Mimi Wang at MD Anderson is using FMT to treat immunotherapy-associated colitis. But again, fecal transplant is hard to really do in some ways, and it's not scalable. And so what can we do? And so what we've actually done is we've actually now taken one of those donors who had a complete response to treatment with immunotherapy. Her fecal sample was then used to treat other patients who then had responses. And we actually engineered a synthetic community of microbes to the tune of around 60 different species with Canvas Bio, where we actually now have this defined consortia, which we can grow at scale. Actually going to be submitting an IND for this and taking this into clinical trials within the next year and a half or so. And so, and what we've, so we now are engineering these synthetic community of microbes using bugs as drugs and using also targeted antibiotic approaches to improve outcomes in patients with cancer. So a little vignette is, a couple of years ago, Susan Bowman, who was at the Hutch, is now at MD Anderson, showed in a publication in Science that treatment with a targeted antibiotic approach was actually associated with delayed tumor outgrowth and in colorectal cancer. And it was metronidazole, and it actually was depleting fusobacteria from the tumor microenvironment itself. Well, we were intrigued by that, and we actually looked in preclinical models and treated mice with metronidazole and showed that it actually augmented response to immune checkpoint blockade. So could it be that simple? We then looked in a clinical cohort, found that if patients had actually seen prior treatment with metronidazole or Flagyl before going on to treatment with checkpoint blockade, they actually had much better survival. And we're now validating this in additional cohorts, and we have clinical trials underway. But wouldn't that be interesting if it was that simple? And what we see in the gut microbiome of those treated mice is that you actually get a reduction in the unfavorable taxa, Prevotella, and some of the other bugs, and you get a bloom of all the response-associated microbes. And so I think opportunities certainly to collaborate, to do this together, and to really understand mechanism, as Barbara was saying, to really drive this forward and identify new targets. Now, how about the probiotics? And so we actually, in a follow-up study to our original publication in 2018, we actually asked our patients if they were taking over-the-counter probiotics, because an unintended consequence of really doing these studies is that everyone wants to run out and modulate their own microbiome. We found that, lo and behold, 30% of our patients reported taking probiotics off the shelf. And if they did, numerically, they actually had worse outcomes. And so We were intrigued by this, took it back to a preclinical model and took germ-free mice, implanted a perfect microbiome into those mice from a complete responder donor, then gave them either one of two probiotics we bought off the shelf at Walgreens, and then treated them versus sterile water as control, implanted tumors treated with checkpoint blockade. And if they got either of the probiotics, their tumors grew rapidly and they failed to respond to treatment with checkpoint blockade. Okay, what were those probiotics? Align and Culturelle, so bifido and lactobacillus based. So, and interestingly, Lorenzo Cohen, who's the head of integrative medicine at MD Anderson and author of Anti-Cancer Living, co-PI on all these studies, and during the course of treatment, he wrote this, he's been very public about this, he actually developed metastatic melanoma himself, and actually asked us to sequence his microbiome. It was the best microbiome I've ever seen. He actually started to take over-the-counter probiotics, had us resequence his gut microbiome. It looked to be dramatically worse than what we would consider to be, but again, to Aaron's point, or to Arun's point, what's a good versus a bad microbiome? But nonetheless, he was actually able to stop taking those over-the-counter probiotics and get back on his good diet, and actually he's doing great. So, but he put this op-ed out in the Wall Street Journal on those probiotics may actually be hurting your gut health. And so I think we do need to think a bit more about this, and some strategies that patients are using may not help and may even harm treatment. And so we currently recommend that patients don't take over-the-counter probiotics. And we are using strategies to modulate diet and other lifestyle factors, using dietary intervention with high fiber and other diets. And we heard a little bit about fiber and the beneficial effects on the ovary. Well, it actually has a great effect on immunotherapy response. And so we asked patients when they were coming in, what were they eating? We used first a dietary screening questionnaire. We're now doing 24-hour recalls and some more elegant means of measuring the diet. And what we found is that patients who reported sufficient dietary fiber intake of at least 20 grams per day were 5 times more likely to respond to treatment with immunotherapy for cancer, okay? Published those findings in Science in 2021. And then we said, well, can we actually use diet as a precision intervention in cancer, with the hypothesis that a whole foods-based fiber-rich diet will actually modulate the microbiome favorably from a functional standpoint and enhance systemic and anti-tumor immunity. So that's exactly what we've done. So at MD Anderson, we have a bionutrition research core, or a food kitchen, where we actually make meals. We have chefs, they're delicious. We get people up to 50 grams per day, okay? And actually we can package these meals and ship them really anywhere. And what we've been able to do is run several different phase clinical trials, where first in melanoma survivors, next in the setting of treatment with anti-PD-1 or some of the other immunotherapies, and finally with behavioral intervention and diet studies. And what I can tell you is we're seeing really, really dramatic results. But what does it take to get 50 grams a day? So I can tell you, when we asked our patients, and even when we asked our workforce, how many people, or what proportion of people get at least 20 grams per day? Not many, okay? It's less than 30%. So most people hover around 15 grams a day, and it's nowhere near what we need, okay? How do you get to 50 grams a day? And feel free to take a picture if you want. This is, I put this together from this book, Cook for Your Gut Health. But it takes intention, right? You really need to be intentional about this. An apple a day ain't gonna do it, okay? 4 grams of fiber with the skin, right? You can't cut the skin off, can't cheat. But if you look at lentils, black beans, limas, chickpeas, chia seeds, you can get there, but you have to really be intentional about this. I actually have this on our refrigerator at home, so my kids and my family can actually aspire to get to 50 grams a day. Most days we get to 20 if we're lucky, 20 or 30, but it really is important, okay? And it makes a difference. And how much of a difference does it make? Well, let me show you a clinical vignette, and then I'll tell you, hot off the press, not yet published, results from our clinical trial, okay? So this makes a difference every day, I'm telling you. So this is a patient that Jen McQuaid and I treated. So 43-year-old female came in to see me, right, for surgery, because she had several sites of metastatic melanoma in her arm, okay? And she had at least 2 sites of disease. We actually treated her with immunotherapy, with immune checkpoint blockade, also enrolled her onto one of these high-fiber diet trials, and lo and behold, 8 weeks later, I couldn't even feel those tumors, okay? And this was during COVID, the height of COVID. We're both beaming underneath these masks, like amazing. I was so impressed. She actually paid out of pocket for her immunotherapy because it wasn't covered by her insurance. You know how much it cost her? $30,000, right? And so I actually resected these lesions in the clinic. Forgot to bill for it, don't tell my boss. But when we went back, there was actually no viable tumor, right? And she's probably cured, okay? And this is something I would not have seen when I started my career, okay? And we're seeing this consistently, which is really, really great. And this is what we're doing at MD Anderson. So we have this platform or program for innovative microbiome and translational research, or this PRIME-TR, where we have this small army of individuals who really kind of work together. We have a leadership team, we have data science, clinical translation. We also have spatial profiling, a so-called bugs in space initiative, where we're using spatial transcriptomics to look at the presence of microbes within the tumor microenvironment. And really, we're working with the different disease sites, and also within the context of our CCSG in 3 different kind of buckets, if you will. We do microbiome profiling and cohort studies, along with research and development and mechanistic studies. And then now, thankfully, actually getting to a lot more of these microbiome modulation trials. And so, that's really where it's at, I think, and especially, I think, in the short term, as Iran pointed out, I think diet intervention is a really nice way to be able to modulate the function of these microbes and to actually improve outcomes. And I think that's a really excellent place to start. And it's something you can do for your patients, because every time a patient comes in to see us, right, they want to know, what can I do to help myself? It's a pretty good way to help is by just increasing the fiber in your diet, right? And so, I go back to the question again, could microbiome targeting become the next pillar of cancer care, with strategies to monitor and modulate the microbiome to treat, intercept, and maybe even prevent cancer altogether someday? And, honestly, I think, we're here. We heard a little bit from Barbara about ways we can profile the microbes in the lung. They actually are developing these smart toilets where you can sit down and actually it'll profile your microbiome and send it back to your smartphone real time. Jen, lay off the red wine, have a little more kale, right? They have these 3D printed pills you can swallow that'll sample microbes all along the length of the intestinal tract. And then breathalyzers for disease. Alstone has this, measures volatile organic compounds, right? Because a lot of these bugs are really producing these VOCs. And, could we actually have a breathalyzer that could detect cancer or other diseases, and really the functional aspects of the microbiome? And then from an interventional standpoint, thanks to brilliant people like Iran Elenov, we're there too, right? You have personalized nutrition and diet, and really kind of ways that you can actually, in a targeted precision medicine way, right, actually, look at the microbiome and then engineer diets for patients or for individuals, right? So the American Cancer Society has actually come to us to be like, okay, how can we work with food packaging, and servicing to actually improve health and outcomes for patients with cancer, right? And how about your microbiome could even play a role in your COVID-19 response? So that's cancer, but we know now that the microbiome and other factors, not only influence cancer, but they're also influencing other diseases. And what tops cancer is heart disease, right? And can we work together to make changes to promote overall health? So can we actually monitor and modulate gut microbes, diet, and other variables in cancer treatment, and to prevent overall health? You think about the diet in the United States is pretty abysmal, right? Could we actually have policy changes where we can actually change this and actually have a much healthier community and individuals as a whole? And so I think just to wrap up, I think we can help address these issues on a global scale and also as individuals. I think the choices we make every day have a tremendous impact on the microbiome and on our own physiology, but also on our planet. So, and again, it's kind of what this whole conference is talking about and what there was discussions on this morning. Can we actually, move away from this left-hand side of the screen where, a lot of meat eating, a lot of inflammation, a dying planet? Can we move to a healthier lifestyle where we embrace plant-based foods, more fitness and health-centered activities, healthier lifestyles, and improve the health of ourselves, but also improve the the health of our planet. And so with that, I will finish my talk and thank you for your support and for all that you do in this world. And thank you for the kind invitation to be here today.